Hi, everybody, and welcome to the September Plant Physiology Focus Issue webinar. And this month, the topic is on cell polarity. We have three speakers, Sabina, Arif, and David. Our host is Jamie, and our moderator is Yoka. Before we get started, I want to point out that this focus issue is online in the Plant Physiology website. It was edited by Juan, Jamie, Victor, and Jan. And this webinar is being recorded and will be posted to the Plante YouTube channel, where you can find all the other focus issue webinars for plant cell and plant physiology. We'd also like to thank the American Society of Plant Biologists, that is the sponsor of this webinar series. We'd love to have you join us as a member, and there's a code, Presents10, that you can use to get 10% off your membership dues. We're going to have a Q&A box that Yoka will be reading out questions at the end of each presentation from the questions box, so please put your questions there. If you're having any problems, you can email me or try dialing in again. You can come back in if you leave. And so with that, I'm going to turn this over to our host. Thank you.
Well, thank you so much, Mary, for all of your coordination of this great webinar. And on behalf of all the co-editors that Mary introduced, we're really happy to put together this focus issue on plant cell polarity. Obviously, we're totally biased, but we think plant cell polarity is amazing and really moving forward in a very interesting direction. And so although our focus issue doesn't include all of the things that could possibly be encompassed in plant cell polarity, we think there are a lot of really interesting articles promoting or showing a lot of new results from a range of different scientists at all the different levels. And then there's a lot of interesting, updates and review articles that I hope you'll read through and consider thinking about how some different people in the field are thinking about plant cell polarity going forward, right? So we know that plant cell polarity is really important for various biological processes, but we're really, all of us, I think, are really interested in how these polarized proteins, lipids, and other molecules, how they're trafficked to the place where they're localized in the cell, what they're doing there, and how they impact development and physiology. And we think there's a lot of really exciting things coming in the next few years. And I don't want to take up any of the time of the speakers today, because I think they'll be really exciting, and I know they have very nice presentations to show. And so I'll turn it over to Yoka, who will be moderating this session, and I thank everybody for being here.
Hi, everyone. So welcome also from my side to all of you. Today I already will directly jump in and start introducing our first speaker, which is Sabine. She's a professor at the the Department of Developmental or Cell Biology at the Friedrich Alexander University in Erlangen. She earned her PhD on the root development at the University of Natural Resources and Life Sciences in Vienna, where she works on the molecular aspects of cellular morphogenesis in Arabidopsis, and her lab focuses on the mechanism of division plane selection, which she started investigating already as a postdoc at UC San Diego. Previously, she worked as a group leader in the Center of Plant Molecular Biology in Tübingen, where she became interested in the establishment of polarity in pavement cells. And today she will give an update on selected cell polarity in mechanisms in plants. Sabine, if you're ready, you can share your screen, and we are looking forward to your talk.
Thank you very much for this really kind introduction. So let me just, I hope you can see my screen now. Yes? Yeah, wonderful. Thank you very much. So today I want to speak about ROP-regulated cell polarity mechanisms that I work on in my lab and that I have discussed in my review in this focus issue. So tip growing And multipolar cells such as pollen tubes or root hairs or pavement cells acquire their shape during spatially and temporally highly controlled morphogenetic processes that involve ROP-mediated local cell wall remodeling and directional growth. So Arabidopsis pavement cells are actually one great example of polarized growth. They are characterized by their puzzle shape with interdigitating lobes and indentations that actually require coordination of polarized growth by neighboring cells. Pavement cell development has 3 stages. In stage 1, the lobes and indentations are actually initiated, and in the following 2 stages, they become more pronounced. 2 antagonistic polarity signaling pathways are responsible that involve small GTPase row-related of plants or short ROPs. The lobe formation is facilitated by the interaction of ROP2 with its effector RIC4. And this is actually responsible for the assembly of F-actin in the forming lobes, and this is promoting outgrowth of this region. In the indentation regions in the neighboring cells, ROP6 is interacting with its effector RIC1. And there, this interaction promotes the bundling of microtubules, and this is suppressing growth in the end. So ROPs are molecular switches that cycle between an inactive GDP-bound form and an active guanosine triphosphate-bound form. During developmental processes, for example, active ROPs interact with their effectors, and that leads to the reorganization of trafficking and the cytoskeleton, as well as cell wall remodeling, and that leads to local cell wall changes. There are 11 ROPs in Arabidopsis. They are activated by guanine exchange factors, or GEFs, and these catalyze the exchange of GDP to GTP. GAPs stimulate the hydrolysis of, GTPase activating proteins called GAPs are then actually stimulating the hydrolysis of ROPs and inactivate these ROPs. So when we started to work on GAP proteins, it's actually a pair of GAPs that we call pH GAPs because of the presence of a plextrin homology domain. There was actually little known about the inactivation of ROP proteins, except for the work on RAN1 from Shenbiao Yang's lab in pollen tube development. So we decided to generate a double mutant, and these double mutants had a very dramatic loss of pavement cell shape complexity involving these proteins in pavement cell shape establishment. So this, of course, raised the question, which ROPs might be inactivated by these pH GAPs? And therefore, we performed protein interaction studies. So in a split ubiquitin assay, both pH GAPs actually interacted with ROP2 and to a lesser extent with ROP6. In addition, we performed co-immunoprecipitation, and we could co-immunoprecipitate ROP2, but not ROP6 from plant extracts that were also expressing pH gap 2, of course. So that led us to conclude that ROP2 is the target of pH GAPs. So of course, we were also interested in the localization. And when we had a first look on GFP fusions of pH gaps, the distribution on the periclinal cell walls of pavement cells was not very exciting, actually. But when we looked at the top views of anticlinal cell walls, we did see some heterogeneities. And in 3D reconstructions, we could actually see this very interesting, distinct localization in stripes of pH gap signal. So when we took optical sections that were slightly above the transition of the anticlinal to periclinal cell phase, we could also determine that the gap signal was located at the indentation sites of cells. So we next asked whether the polarized localization of these pH gaps correlated with lobe and indentation forming regions. And therefore, we used a tool from Dan Szymanski's lab that allowed us to correlate the curvature of cell wall segments with the distribution of our pH gap signal. And so what the tool does is when we select a region, it creates a plot profile of the signal distribution along the anticlinal phase. And so we then quantified the signal peaks or the frequency of the pH gap signal peaks in the apices, the flanks, and outside of the curvatures. And we found that more than half of the peaks were actually present in the apical regions. 30% were present in the flanks and 14% were present outside of undulations. So basically, pH gaps are predominantly localizing to the apical regions of these undulations. Because the pH gap signal along anticlinal cell phases was also reminiscent of microtubule signals that have been observed in along the anticlinal walls, we performed colocalization analysis. And there we found that between 70 and 80% of the pH gap signal also colocalized with microtubules. And we also found that the localization was dependent on microtubules when we did inhibitor studies. So why do the pH gap mutants actually have this remarkable polarity defects? So based on our findings, we concluded that pH gaps localized to the indentation sites of undulating regions, and they persist there to inactivate ROP2. So basically in locations where pH gap abundance is high, the activity of ROP2 should actually be at its minimum. And this then allows for the reorganization of the cytoskeleton and in consequence also cell wall remodeling at the indentation site. So in addition, we also found abnormal cell wall patterns in the root meristem in our pH gap double mutant. So we found elevated frequencies of oblique cell walls, and these tilted cell walls are actually consequences of defects in division plane control, and they reflect either a disorganization of the preprophase band, which is the structure that is predicting the future site of cell plate fusion, or these defects are due to misguided phragmoplasts, which are the cytoskeletal structures that are guiding the cell plate to the cell plate fusion site in cytokinesis. And so therefore, we evaluated the orientation of both of these arrays in our double mutant and asked how much they deviate from the ideal transverse orientation. So indeed, we found a deviation of the preprophase band and also deviation of the phragmoplast roughly to the same extent. And this finding was very interesting because it really suggests that the position of the PPB is compromised and that ROP-mediated cell polarity defects may affect division planes also in symmetric divisions. So to understand the mutant phenotype that we found better, we decided to look at the localization of these pH gaps during cell division. And what we found were these distinct polarized rings in dividing cells. Further studies actually confirmed that pH gaps are part of larger protein complexes that reside at the cortical division zone, which is a polarized region of the plasma membrane that is serving as a memory or as a coordinate system for the division plane throughout cell division. And this cortical division zone is utilized by the phragmoplast at the end of cytokinesis for cell plate insertion. So these observations posed the question how ROP signaling interferes with division plane orientation in symmetrically dividing plant cells. So along with other proteins, these pH gaps are polarized at the cortical division zone. And there it might be required to set up and maintain a region that is called the actin depleted zone. And this is potentially by analogy to its function in pavement cells. So pH gaps keep the activity of not yet determined ROPs at a minimum within this region during cell division. This is a hypothesis that, of course, awaits further evaluation. Interestingly, the localization of ROP GAFs in phloem progenitor cells is complementary to the one that we observed for pH gaps, and mutants in the respective ROP GAFs also have division plane defects in phloem tissue. So to summarize, the inactivation of ROP-mediated cell polarity signaling appears very important for cellular morphogenesis. And the following model that I want to show you is integrating data from my lab as well as from several other labs, and they are referenced in my review article. So local anisotropy in cell wall composition leads to the activation of ROP6, for example, via Feronia kinase, which phosphorylates ROP-GAF14, and that activates ROP6. This leads to the ordering of transfacial microtubules along the anticlinal cell faces. And these feed back on the cell wall via local cell wall reinforcement through the parallel alignment of cellulose microfibrils. On these progressively indenting regions, these transfacial microtubules may also promote the recruitment of pH gaps, which then in turn deactivate ROP2, and the stabilization of pH gaps along these microtubules is likely accomplished by PIN2 kinase. On the other hand, at the opposite lobe site, brasinosteroid signaling inhibits spin kinase, and this destabilizes or reduces pH gaps, which allows then for the activation of ROP2 in the slope forming region. And then there's accumulating evidence that ROP signaling is also involved in symmetric division, but this hypothesis, as I said, requires further studies. And with this, I'm at the end, and I want to acknowledge my fantastic team that has done all the work that I presented, great collaborators, and my funding bodies. Thank you for your attention. And
Thank
I
you,
need
Sabine, for your...
sorry. Yes, I need to stop.
Okay,
sharing.
while you're stopping.
Yeah. Okay.
Thank you for your interesting talk. I think this was a very good start of this webinar. So there are already some questions that came in, and for all those that are still typing the questions, please go ahead. I will start with the first one, which is a more general question. If the lobes and the indentations of the pavement itself serve any specific purpose?
Yeah, so that seems to be to reduce mechanical strain within the tissue surface, basically. So this is the current hypothesis.
Mm-hmm. Then do you know if the ROP2 and GAP1 localization are similar or not?
So it hasn't been shown for ROPs that they are localizing in those stripes, although I have not seen any study that has looked at the anticlinal cell faces. But within the periclinal walls, the ROPs, this has been shown by super resolution and turf microscopy, are actually localized in polar domains that are made up of nanodomains. And these nanodomains are heterogeneous in composition in terms of phospholipids as well as proteins.
Okay. Mm-hmm. Next question. Does the pH GAP1-2 mutant has defects in the maintenance of PIN2 and or PIN1 polarity in the root?
So we did look at, did we look at that? So yes, we did look at that, but maybe we haven't looked in enough detail to see any differences. So by the time we were looking at, at the PIN2 localization, it appeared fairly normal, but maybe the abundance is reduced. So we haven't looked at that in detail.
Okay. And is there a difference? So now you focused on pavement cell and roots, but were there also different physiological changes in transgenic plants?
So we have not tested that. We have only tested whether the mutant is actually has any differences in water uptake, because stomata opening and closure is also regulated by ROPs. And there we did see some changes in water homeostasis in the mutant, but we didn't follow up on these physiological defects any further.
Mm-hmm. And then last question for today. Where would you place actin nucleators like ARP2, 3 in your model?
Well, I would put them definitely. So you mean on the whether it's on the indentation or on the lobe? Well, I would put it on the lobe side. That would be my guess.
Mm-hmm.
But
Yes.
yeah.
Good. Then thanks one more time, Sabine, for your very interesting talk.
Thank you.
So Arif, if you're ready, you can already start sharing your screen while I introduce you. So Arif is a plant biologist, and his lab at Howard University utilizes Arabidopsis and maize to study nuclear envelope proteins during movement and mitosis. Previously, he did his postdoctoral work in the lab of Michel Facet on polarized nuclear movement during asymmetric cell division. And during his graduate study in Abidur lab, Arif first started working with polarized proteins and studied polarly localized transporter proteins during plant development and environmental response. Further, Arif also hosts a plant biology podcast called No Time to Read, and he is co-founder of the Plant Postdoc community. Today, he will talk about the nucleus decides the future cell division site. Arif, if you're ready, you can start.
Thanks for the introduction. Hi, everyone. I'm Arif Rashaf. I'm a very new assistant professor at Howard University. So what does it mean? I just joined here a month ago. So the work I'm going to present here today is actually started from my postdoctoral work with Michel Facet at the UMass. So I'll start with something very, very basic. Like when you think about a multicellular organism, we basically see that it becomes from one cell to many cells. The obvious question is how it became. We know very simple thing that when the cell increases in number, it goes through the cell division. But also along the way, we see that it's not only the number increases, also we see one cell type, and then it becomes many, many different cell types to function very different things. So how that thing happened? So that also happens to cell division, but very specific kind that's called the asymmetric cell division. So for every multicellular organism, the idea is that when you see many new different cell types doing different function, it is actually coming through the asymmetric cell division. So if asymmetric cell division is that important, let's just focus on what is fundamentals different about asymmetric cell division first before going into the details. So usually all of us study the cell division in the very textbook thing in the classroom. And what we learned that, okay, one mother cell divides and produces two daughter cells. And here both cells look like same size. And also the same color code indicates that their cell type will do the same function. But here is the problem. If we have only this kind of cell division in life, we'd never come up with a different cell type doing different function. So in biology, we see a very different kind of cell division, which we call the asymmetric division. And what it does, that one mother cell divides and it produces two daughter cells. They look like same size, but with a different color code, I'm actually indicating that they have different function. But to understand that they went to the asymmetric division, you really need to know their molecular marker of these two daughter cells. But there is another kind of asymmetric cell division when the mother cell actually divides. We can see there are two cells. Immediately you realize that they have very, very distinct size, and also with the different color, I'm indicating that they have different function of cell types. So in plant tissues, we see this kind of, the third one, the distinct volumetrical daughter cell kind of asymmetric division a lot. So here is an example. In different plant tissue, we see this kind of, the third type of second type of asymmetric cell division. And what happens inside the cell knows the direction based on the polarized protein localization. And let's say, based on that, the nucleus moves to the one side of the cell. And once they divide, they produce one small cell and one bigger cell. And who moves the nucleus in this case? So here I'm using the same figure, except I'm just color-coded with either it is microtubule dependent or actin dependent based on all of the fantastic work happened before. And for today's talk, actually, I'm going to focus on not many cell types, but also for the maize stomatal development, or what we're seeing here. So I showed you enough cartoon and told you what is asymmetric cell division. So let's try to look at how these cells looks like in the plant itself. So this is a corn plant. And if we want to see that how these cells actually go into the division and eventually become what they become, so we can take actually just one single leaf. And if we look at very early stage of the development, we see there are a bunch of cells, all of them look like kind of same, we cannot tell them apart. But once we go further down the development stage, we see the cells started looking very different from each other. That is the time actually they're going to have their different identity. And once you go even further, what you see that the cells have a very characteristic shape. By this time, they're differentiated and doing their specific function. And among all these cells, we're actually going to focus on the cells here. Probably you'll see a very tiny hole, and that is the actually stomatal pore, which is maintained by some dumbbell-shaped cells or the gut cells open and closing. And just adjacent to them, there are two other cells on the both sides that are known as subsidiary cells. Why you are talking about these cells? Because this is one of the cell types of the subsidiary cell is actually produced through the asymmetric cell division. Now we know what is asymmetric cell division, what kind of cell types we are going to look at. Let's see things more into action, what happens inside the cell. So in this movie, what we are going to look at in this cell, basically this magenta colored protein is a polarized protein. Based on that, cell knows the direction. And if you see the cell division, it happens on one side of the cell, and it produces one small cell and one bigger cell. That is the fundamental kind of asymmetric cell division we're going to talk about today. But this movie is great, but what you don't see the nucleus in this movie. So then the next movie, I'm going to show you similar thing, only you can see the nucleus. So among this whole area, these cells, you'll see this nucleus is polarized in this side. And when the cell divides, basically it produces one characteristic small cell and one bigger cell. And what actually empowering us using this system, because we just can look at the cells and can understand whether the cell division goes right or wrong. And so what is the question we're trying to answer here? Basically, we know a fair amount of work about the cell polarity, and also we know a lot about cell division. But during this kind of asymmetric cell division, the nucleus needs to move to a certain direction. And this movement is actually mediated by the cytoskeleton, that is also we know. But the way classically those studies are done, basically we try to inhibit using the drug all the cytoskeleton in the whole cell. On that case, it not only affects the nuclear movement, but also some other cellular things as well. So we try to look at very carefully on the nuclear membrane. And as we know, it's a double layer membrane with inner and outer nuclear membrane. And there is some protein complex called LINC. They basically maintain the link between inner and outer and the cytoskeleton in the cytoplasm. So what we try to do, we actually targeted one of the outer nuclear membrane protein, part of the LINC complex that maintains this cytoskeleton and the nuclear interaction. And this protein is actually called MACE, link cache complex. You don't have to remember, it's a fancy name, it's fine. So we're just going to talk about one of the outer nuclear membrane protein part of the LINC complex. So the idea is that if we disrupt the link, the nucleus and cytoskeleton, probably the nucleus will not be able to move, and we can actually use it as a tool to answer some of the questions, how nucleus actually, or nuclear movement plays a role in during the asymmetric division. So the first thing first, if we use that mutation, as I told you, that we can just look at the cell shape and tell that whether the cell division goes right or wrong. So here, anything you're looking at the cyan, basically, if the cell asymmetric cell division goes right, anything with the magenta didn't go well. So these are all the terminal cell division defect you can see and we can quantify. So it tells us very basic and a very simple thing that this protein on the outer nuclear membrane, which maintains the connection with the cytoskeleton, is actually required for asymmetric cell division. That's okay. That's the first thing we came to know. But one of the important things we want to know, okay, this cell division, terminal cell division defect happens, how it is happening inside the cell. So because of the nature of this mutation, we disrupted the link between nucleus and cytoskeleton. So we wanted to look what happens with the nuclear migration in that context. So if this is my polarized side of the cell, the nucleus is supposed to come and touch this side and become polarized. If nucleus is almost there, not polarized, we're calling it offset nucleus. Or nucleus can be farther away from the polarized side, which we are calling the unpolarized nucleus. And we can actually go through those cells by tagging protein, which actually shows the nuclear envelope, and see where the nucleus is in a particular developmental stage. And if I just summarize all these things, what we will see that these two things happen in the higher frequency in the mutant, like offset and unpolarized nucleus. Should we be surprised? No, because this is the nature of the mutation or things we're dealing with. Rather, it actually helps us because the pre-mitotic nuclear migration or position is altered. So we can actually use that as a tool to ask, okay, what happened if nucleus is not the correct spot to the cell division? So here we actually are very grateful that plant has something called the preprofess band. So the preprofess band is something even before going to the division, cell actually mark with a microtubular ring that where they will divide, right? Division site. So in short, it's called the PPB. So what we're thinking that, okay, we can actually try to look at where is the nucleus and where is the PPB or future division site mark in the same cell. So in this picture, what you're seeing, anything with the cyan basically indicates where the nucleus is, and also anything with the magenta indicates where the preprofess band is. So if the nucleus is polarized, nice, the preprofess band is the correct spot. But what we started to observe that if the nucleus is not in the right spot, let's say offset here or unpolarized, we start to see that the preprofess band formed transversely. Is it a random event, or is it correlatable with the nuclear position or the PPB? So for this, we did very nitty-gritty quantification. And the basis of that, we looked at in the same cell, where is the nucleus, polarized, offset, unpolarized, and at the same time, where is the PPB in this cell? And usually what happens, if the nucleus is polarized, the PPB forms in the correct spot. And regardless of wild type to the mutant, we see it from 80 to 90% cases. Sometimes you see the nucleus is almost offset, but the PPB forms in the correct spot. But in this whole quantification, when you go cell by cell, one of the things we need to focus on is basically once you see the preprofess band form transversely, either we see the nucleus is offset or unpolarized. And if we add up all this number, it actually goes to 8 to 10%, which is the terminal cell division defect we see for this mutant. Which is okay, because we can see that nuclear position and the PPB position, the transverse orientation actually kind of correlate. But here is the thing, is it the nucleus and the PPB decides and all the mitotic steps follow that? Or like some individual mitotic steps can go wrong independent of the preprofess band and nuclear position? And we can tell this, and only way we can tell this, if we can capture the things in the cell from the very beginning of the cell division to the very end. So we went and did. What we did here, basically, if I play this movie, the nucleus is polarized in the beginning, and these two bright spots indicates the preprofess band. And once the envelope breaks down, the spindle forms, fragmoblast, everything goes according to the initially marked. I'll let it rewind to play this movie because this movie is a reference one and very important one. Nucleus is polarized and preprofess band in the correct spot. Once the envelope breaks down, the spindle forms, the fragmoblast and everything connects, and we get one small cell, one bigger cell. That is nice and excellent. But the question is, if nucleus is not in the right spot, what happens? So in this movie, we're going to see the nucleus is offset, and we see these two bright spots indicates the preprofess band, which is transverse. And once the envelope breaks down, actually the spindle and the fragmoblast connects where the initially preprofess band marked. So let's watch this movie again, because this is very important. Nucleus is offset, preprofess band is transverse. Once the envelope breaks, the spindle forms, but they actually orient based on where the initial preprofess band mark, and all the mitotic steps follow that. These are not the only one or two movies. We went hours after hours, months and years to capture the cells dividing from the very beginning to the end. And these are the snapshot of the two movies I showed. And if I boil down all of the many, many hours we went to the confocal imaging and the live-stream imaging, basically we come to the point, if it is wild type or mutant, if the cell division goes right, we see at the beginning, the preprofess band was marked correctly. And all of those cells, most of those cells actually has a polarized nucleus, which indicates P. But if we see the cell division goes wrong, we see at the beginning, the preprofess band was actually in the transverse orientation. And guess what? In all of those cells, either the nucleus is offset or unpolarized P. So with this, basically we come to the point that initial nuclear position is actually helping to decide what the future cell division should happen to the PPB and all the mitotic steps happening there. And the very simple model out the very basic biology learned from this is that if in this kind of asymmetric cell division, if the nucleus is polarized, the cell marks the future division site with the preprofess band. And when the envelope breaks down, the spindle, fragmoblast, and cytokinesis follow that. But if the nucleus is not polarized or in this position, basically cell marks the future division, according to nuclear position in a transverse orientation. And on that case, when the spindle forms, fragmoblast and cytokinesis actually follow that. So with this, I actually want to thank my postdoc advisor, Michelle, who played a very instrumental role during my training. I want to thank her a lot. And also all the FAST Lab members, because they are not my lab members, they are my friends and family. I'm really grateful for them. And also I want to thank all the federal agency support and the university level support till today. And with this, actually, I'm a little bit heartbroken because it's very hard to put together all my movies into 13 minutes. But there is a major chunk of the other movies you'll find in the paper. But before I go, if you forget about today's webinar, about me, about everything, I want to I want you to take one-line message from my talk that nucleus decides the future cell division site. If you forget everything else, you should remember that. With this, I would love to take any question. Thank you so much.
Thank you, Ari, for your wonderful talk. I think we're all looking forward to see the first research of your own lab.
Me too. I'm very excited for that.
So the first question is if you could speculate on what is the initial trigger for cells to polarize. And not only in tissue context, but also like cell signaling to the neighboring cells. What would be the initial trigger here?
Okay, so we're talking about very, very upstream. So like the first of all, the cell got some polarity to the polarized protein, and that signal can be intrinsic or extrinsic. So far, what we know, we discover over the many of years of work of the Laurie Smith or Michelle or the other people that we identify some of the polarized protein, and we know that timing inside that cell. But we really so far don't know, okay, what is the, because most of those are like receptor kinase. We don't know whether they have any ligand so far. So extrinsic factor, so far we don't know, is still yet to discover. But the intrinsic factor also, we know basically the hierarchy of the who comes first or what comes next. But we really don't know like what is before that.
Mm-hmm. Next question. Is there any evidence that cells do asymmetric divisions as in exocytose, like disposing a content that is less favorable in any reason or for any reason?
I think I didn't get the question very well. If the question can be expanded, that would be better for me to answer.
Let's have a look. In the meantime, I ask next question,
yeah,
and
sure.
he or she can elaborate on the question. So how is nucleus regulated by PPB position?
Oh my God, this is my favorite question. Yes. So one of the things we actually can look at right now, because the kind of figure I showed, I can look at the midplane and the cortex. And one of the things that question answered, I also have the same question, like to see not only when the preprofess band already formed and the nucleus is there, but also the formation of the preprofess band when the nucleus is the right spot or not. So I think like there are some challenges on how we see things. So anything I did with this stage or all the materials, basically I used spinning this, so which is great for the time-lapse imaging and long-term imaging. But I really want to go up to like using Airiscan or something where I can see the cortical microtubule formation in very nice and crisp, and at the same time do the time-lapse. So I can see if the nucleus moves, what happens to the preprofess band? Did they dynamically decide to move somewhere and start forming? So this will be like one of my most favorite questions. And literally I'm planning to ask for an instrument grant to buy an Airiscan. So let's see how it goes. But this is a fantastic question. I think this is one of the questions I would love to see either I answer it from my lab or someone answers it.
Great. Then next question. Are PON1 and 2 regularly polarized in your mutants?
Oh, yes. So this is the thing that what happens when you cannot fit together all the data of the presentation. So I'll just quickly show you. It's actually in the supplement. If you go to the paper, you'll find out. So yes, we did look at, and we spent quite a lot of time to look at all the known polarized protein like BRIC, pen to pen one. And no matter what happens, if the cell division happens right or wrong in the different stages, like we see the polarity is intact, at least for all of those known polarity markers we have currently available.
Great. And then we got follow up on the question that was not so clear. So does the cell divide asymmetrically to remove some undesired chemicals, resulting on one cell with higher chance of survival?
No, we so far don't have this kind of examples, but sometimes the way the things are drawn or something like that, it looks like it's something going out, but it's not like that. So far, what you know from animal system and plant, basically it's like they separate the phage or either some protein that actually can help them to get different identity. That is so far enough. We don't have any examples in hand that, okay, they're dividing asymmetrically for survival purpose or not. Correct me if I'm wrong, if there are any reference out there.
Great. And for the sake of time, we have to move on to our last speaker, but thank you one more time, Arif, for your great talk.
No problem. Thank you so much.
Our next speaker is David. You can already start sharing your screen. David is a first-year PhD student at the Lab of Integrity Structural Biology at Charles University, and his work aims to elucidate the complex relationship between the evolutionary and molecular architecture of callose synthase and its functional implications during plant development. And before this, he dealt with the characterization of protein players involved in plant signaling, as well as amino acid metabolic enzymes during repeated stress exposure. David is an avid cook, musician, and also an amateur Wikipedia editor. And today he will give an update on the evolutionary insights of callose synthase at the center point of plant development. David, it's all yours.
Hello, thank you. Can you see my screen? So I thank you for a nice introduction. I welcome you to the presentation of our paper, which is titled Callose Synthesis at the Center Point of Plant Development and Evolutionary Insight. If you want to read the paper, just follow the QR code here. So to start, it is now known that the plant cell wall is made up of a mixture of compounds. These include cellulose, the structural scaffold of the plant cell wall, xyloglucans, supporting the role of cellulose, or for example, pectins, acting as a molecular glue. These compounds could be called canonical because they are present during all plant developmental stages. But in addition to these compounds, there are also another compounds, for example, amorphous callose. Callose is deposited polarly in plants in both space and time, meaning that its synthesis is relevant during various developmental stages. For example, in developing microsporocytes, callose separates the individual cells within the tetrad, or for example, it is important for the growing pollen tube. In addition to this, callose also regulates cell-to-cell connectivity. For example, in Plasmodesmata, callose deposition regulates size exclusion limit, essentially inhibiting intercellular transport from one cell to another cell. And this is also relevant for example, phloem development, where callose deposition is necessary for the correct maturation of sieve elements. And also, callose is also important for the development of trichomes, where it can be seen deposited in a so-called Rothmannian ring. However, callose is also important, for example, for pathogen response, where callose-rich papilla is formed in response to pathogen attack, or for example, for the plugging of damaged phloem. In all of these cell types or processes, callose is the secondary compound that deposited into the existing cell wall. However, there is also one cell type or cell process, cytokinesis, the ultimate step of cell division, where callose is actually the first and predominant polysaccharide in the growing cell plate, which grows from the inside of the cell to the outside of the cell. And since there are such marked differences, we will now look at how is it at the molecular level. So plant cytokinesis is mediated by two distinct structures: callose-rich cell plate, which grows from the middle of the cell to the sides of the cell, as shown in this great video, and also microtubular phragmoplast, which acts as a highway for the Golgi-derived vesicles, which contained the material for the cell plate. And their mutual action eventually leads to the formation of a new cell wall, as shown previously. As such, this plant cytokinesis, as shown, for example, in this video, has been best described in land plants, for example, angiosperms, mosses, or for example, in gymnosperms. So naturally the question arises, how is it in their ancestors? And scientists have already looked into this and found out that plant cytokinesis underwent marked changes from throwing yeast, meaning that the Saccharomyces cerevisiae fungi undergoes centripetal cell division, centripetal cytokinesis from the sides of the cells, mediated by cleavage throw, forming the cross wall. And this is also true, for example, for chlorophyte algae. However, in Phragmoplastophyta, they divide by centrifugal cytokinesis, mediated by phragmoplast and cell plate, going from the inside of the cell, forming the cross wall centrifugally. And also, this means that centrifugal cytokinesis must have emerged secondarily during the evolution of plants. And also, it would mean that there should be some intermediate mechanism of plant cytokinesis between the centripetal and centrifugal mechanisms of cytokinesis. However, do we have any evidence of such intermediate cytokinesis? Indeed, we do. For example, in this beautiful paper by Georgia Kakaki lab, they showed that this Zygnematophycean Penium margaritaceum divides by mutual action of cell plate-like structure punctum, and also cleavage throw, as shown, for example, in chlorophyte algae, which are both rich in callose, as shown here by aniline blue staining of callose. And schematically, it looks like this. So we have cleavage throw at the sides of the cell and cell plate-like punctum in the middle of the cell, and they connect to form the early cell wall. These authors have also shown that upon application of endosydin 7, a specific inhibitor of callose synthesis, the callose is totally abolished in these dividing cells. However, the cleavage throw still progresses as in ordinary dividing cells, meaning that callose is significant only for centrifugal cytokinesis. Centrifugal cytokinesis, remember, present in Phragmoplastophyta, going from the inside of the cell to the sides of the cell, which are present in gymnosperms, mosses, or angiosperms. So this is indeed interesting, and in our lab, we asked, could callose synthesis be the driving evolutionary force behind centrifugal cytokinesis? And we wanted to take a closer look at this by analyzing callose synthesis, which mediate the deposition of callose in plants. These are large integral membrane proteins, which are multigenic, essentially meaning that there are as many copies in various plants. For example, there are 12 copies in Arabidopsis. In addition to this, callose synthase is a great subject for phylogenetic studies because it is ubiquitous in planta. All plants have it. It is fairly conserved. It has a good amount of copies, meaning that there are enough of gene copies in each species, but not too many. And also, there is a contrasting role of callose in centrifugally and centripetal dividing species, as I have shown you previously. So we have done a phylogenetic analysis, which is described in our paper, and we have shown that cell plate-dependent plants, Phragmoplastophyta, form 3 distinct subfamilies, which we titled callose synthase A, callose synthase B, and callose synthase C. And these 3 subfamilies are monophyletic, meaning that they have one common ancestral callose synthase from which all of these 3 subfamilies have evolved. So the question is, what prompted this divergence? Could it be, for example, a result of terrestrialization? Because the experimental evidence nowadays shows that new genes emerged thrice in evolution. This is either in relation to multicellularity, going from the unicellular plants to multicellular plants, during the transition to land, going from aquatic species to land plants, and also specifically for land plants during whole genome duplications. However, our phylogenetic analysis shows that mesostigmatophytes, or you can call them multicellular saprophytes, cluster outside the ABC system, meaning that it could not be related to multicellularity. And we have also shown that at least 3 ancestral ABC genes were already present in aquatic cell plate-dependent plants, meaning that this divergence of callose synthesis could not be related to transition to land or whole genome duplication. So any of these 3 are not a clear explanation of those 3 subfamily system. So the question still stands. Is there any relationship between callose synthase divergence and emergence of cell plate? Because callose synthases have evolved from one ancestor callose synthase, which was followed by callose divergence into the 3 subfamilies, as we have shown. And only after this event, centrifugal cytokinesis emerged, and this was prior to terrestrialization of land plants. So the current hypothesis stands that callose synthase evolution would mean that would imply a cytokinesis specific callose synthase. So we now know that cell plate occurred after callose synthase divergence. We also know that callose is required for cell plate. Remember the Androzyden 7 example in Penium margaritaceum? So the hypothesis is that one of the 3 callose synthase subfamilies had to be cytokinesis specific. And the question is, again, which one? So the current evidence that has been mostly done in Arabidopsis shows that there are several callose syntheses related to cytokinesis. This being callose synthase 1 from Arabidopsis thaliana, whose GFP fusion localizes to the cell plate expressed in BY2 cells, and callose synthase 1 is from subfamily A, but also callose synthase 10 from subfamily B, which again has a cell plate localization, and also callose synthase 10 mutants have impaired cytokinesis, which leads to the seedling lethality. In addition to callose synthase 10, another paralog from subfamily B, callose synthase 9, it is gametophyte lethal when mutated. And this is related to cell division, as can be seen here, where the mutant, when the plants deficient in callose synthase 9 don't form this beautiful aniline blue cross wall as in the wild type. And in our paper, we have also shown that in subfamily A, it is the only subfamily in throwing Klebsiella, and also that there's a history of secondary losses throughout plant lineage. And as for the subfamily B, our data indicates that in Arabidopsis, both proteins are related to cell division, and also that they are present in all cell plate dependent species in at least 1 copy. So to summarize, callose plays a different role during cytokinesis of plants depending on either cell plate or cleavage throw, and from this, a question emerges, is callose essential for centripetal septum formation? And also, callose synthase divergence into the 3 subfamilies occurred before the cell plate, as we have shown. So the question is, could the callose synthase duplications play a role in establishing the cell plate? Or for example, does any callose synthase subfamily possess the ancestral role in cytokinesis? And these are questions to which I would really like to find answers. And also, that's all from me. Thank you for tuning in. I will also like to thank my collaborators, Roman Pleskot, and our evolutionary guru, Samuel Halushka from Laboratory of Cell Biology, and also for the organizers for organizing this great seminar. And thank you.
Thank you, David, for your great talk. While people are still busy writing their questions, I have a question. Do you know what regulates the callose synthase activity?
So there is not much data on this actually, but data from our labs indicates that it could be, for example, related by specific phospholipids at the membrane. And also there are several interactors of callose synthase, for example, UDP glucose transferase, which is believed that it feeds the UDP glucose precursors for the synthesis of callose chain into the extracellular space.
Mm-hmm. Is there any experimental accessible species with single callose synthase B?
Yes, there is. For example, Marchantia polymorpha, the liverwort, has a single callose synthase A subfamily paralog, single callose synthase B, and callose synthase C. So it looks that it will be a nice experimental model.
Mm-hmm. Great talk, David. Would these evolutions and duplications be involved in or associated with the evolution of embryogenesis?
That's a really good question. Actually, there is no data currently on this because nobody has really described callose deposition in cytokinesis in embryos. So currently, I can't really answer this question.
Good. Then maybe still one question from my side. Could you kind of speculate what could like polarize this callose synthase?
That's again a good question. I have mentioned the specific phospholipids, membrane phospholipids, and actually, for example, the cell plate is contains specific phospholipids. It starts with, I think, PA, phosphatidic acid, and some of our data indicate that phosphatidic acid could bind calosyntase. However, it could also be regulated, for example, by specific interactors. However, currently there is no evidence into this. Actually, to elaborate further, I'm not sure whether there have been many papers describing the specific polar trafficking of calosynthases, and this is really an intriguing field to find this out.
Yeah, I agree. If there are no further questions, I think we can wrap up and thank all the speakers one more time, Sabine, Arif, and David, for your great talks. Thank you very much. And I give the speech again to Jamie.
Wonderful. Thank you guys so much for all those wonderful talks. And I hope the audience was excited and motivated to go and read the papers in the focus issue. And so before we close, I just want to thank all of you for your really nice presentations, and to the audience for their really great questions. Yes, wonderful. And then again, on behalf of the other editors, Yuan, Jen, and Victor, we really enjoyed putting together this focus issue for all of you to read, and we express our deep gratitude to all of the authors who put a lot of work into their publications and their papers. And we sincerely hope you enjoy reading them as much as we enjoyed putting all of them together. So thank you all for your time this morning, and we hope you have a good day or evening, depending on where you are.
Thanks, everybody. This was terrific. I really enjoyed those talks. See you next time. Have a good evening. Bye. Thanks.
Thank you.
Bye. Thank you.
Bye.